right, so how many of you teach science? Okay. How many, let's just go ahead. How many years you been teaching?
2 years full time, and did chemistry for one class of chemistry, so 3 years in a row.
So 3 years. What did you do prior to that?
10 years as a youth pastor.
Okay. All right.
But I was a bachelor's of science in chemistry, so I went chemistry. I was in the lab for a little bit, and then went to youth ministry, and then now I've come back to the class.
So you had a pretty deep background in it.
Chemistry, yeah.
Okay.
Yeah.
How about you guys?
I've taught for 31 years.
31 years? Okay, I'm not going to be intimidated by that. All right? I won't. All right. So 31 years of teaching science, what does your labs look like? I mean, can you walk us through the kind of processes you see and you use for a lab?
I try to be hands-on as much as I can. My philosophy is a Chinese proverb that says, I hear and I forget, I see and I remember, I do and I understand.
Yeah.
And so I will, if my best class is where I am going from lab to lab to lab, and then the book follows along. So that's what I try to do.
That's where I'm going for.
Yeah.
That's where I'm looking forward to. So there's lots of things that can affect how you do labs, the infrastructure that you're working with, the time period that the class falls on, whether you can make something fit within that. How about you?
Only 2 years I've been teaching science, but I'm homeschooling my own kids.
Okay.
So
Then you have a competitive advantage because you can schedule the time and you can walk through this process I'm going to share today really slowly. So my name is Faron Usrey. I have been teaching science for 3 years here at the School of the Ozarks. I was 24 years with the federal government doing science work. I was an aquatic ecologist at Buffalo River for about 10 years, and prior to that I was a hydrologic technician, collected other people's data. I came to classical education through homeschooling. My wife, who's here, not in the room, but here in this place, she came to me with this idea of teaching homeschooling, and I was like, we both have jobs. Tell me how you think that's going to work. But she was a physical therapist for 15 years, and then she started teaching at the community college. She taught intro to biology a couple years, but her main thing was anatomy and physiology. Now she's teaching the advanced classes. So she was like, it's not working because the students coming out of public schools can't function in my classrooms. And of course, she gets judged on the quality of the work that her students do as far as what their grades are. So she was interested, and plus she had come across maybe Dorothy Sayers. I'm not for sure exactly what got her in that process, but she just come to me and she says, I am convinced this is the proper way to teach, and not only that, it's the natural way people learn. So I have found some things, and I'm not saying that I generated these. I just was in communication with other science teachers. They let me rummage through their toolbox and said, hey, I like this. This will work, you know. And so it was more of a pragmatic approach. How about this side of the room? How long have you been teaching in science? Yes, ma'am?
I've been teaching for 10 years, but I'm in my first year of actually teaching science.
Okay. So how's that transition going?
It's actually going better than I thought it would.
Okay. So my first year, I was literally 2 weeks in front of my students, just trying to get everything in order. I didn't have my teacher toolbox. I didn't have my lexicon out there. I didn't have a lot of vision as to how to make this thing enjoyable as well. And so I was pulling on things that I had learned from what I enjoyed in school until I slowly come into this idea and understanding of a little bit more what it means to teach classically. I think somebody else had their hands up over here. Okay. Well, one of the goals that I'm trying to transition into is just not what we're doing here. I mean, what we're doing here is that you're being a passive learner, and I'm up here the one trying to impart the knowledge. I'm the one trying to bring the organization and energy into the conversation. And the idea is that we need to transition from that passive to where it's more active learning. Now that's difficult. There's been some studies to back this up, and I'm not going to quote any because I'll end up getting it wrong, but basically there was a study group that happened where they did active and experiential learning, and then they just did lectures over here. And the ones that did the experiential learning had much higher retention rates and testing rates, but their perception was that they didn't learn as much because they're so acclimated to this passive learning style. And plus, it makes it more like an entertainment to where their mind is not actively engaged in assessing a problem or diagnosing an issue or formulating an argument. So the labs that where I'm trying to create as many as possible puts the student into the driver's seat. I remember doing labs quite a bit. You would come and go to chemistry lab, you'd be handed a sheet, and if you read it or not, that was, some people never seem to do that, but you're usually on a team of people. You're given these instructions, and a lot of times the labs that I had were not connected with the things I was learning in the classroom. It would be almost a completely independent concept that might come into play a couple chapters later on, but it worked with the lab schedule. And then a lot of times it wasn't the same teacher. It was the lab technician running the lab, teaching you that concept. And so for me, labs were always like, I hate them things. It's chaos. There's people rummaging around looking for things. The directions do not make sense because I do not know the terms. So first time I'd seen the terms is when I've got the handout. So there's a lot of frustrations with the traditional way of doing labs. It's not a bad thing that students have to read and follow directions. So I'm not saying that, but you so much can lose the actual objective of the lab if you approach it in that way. And I still do some labs that way because of the time period that I might just have a period, a block period, or just a single period to make this point. And so it turns to be a little different. So I think that these the student-led scientific inquiries, it puts them in the driver's seat doing science. You know, we have to lay a foundation before they can do that of the grammar and the logic, but they are the ones who are practicing science. Each one of these labs, they have to go through the scientific method and understanding what inductive reasoning and deductive reasoning, what parts they play in the scientific method. It's not just inductive sometimes. So I do think that this is a very classical approach, and I'll give my reasons for that. So this is, again, based upon the scientific method, and it's not, in the past, when I was in classes or I'd taken classes, they would teach the scientific method at the very beginning, maybe chapter 2, and you would never hear it again. So each time that one of these labs you do, you go back through the scientific method and you start practicing applying that procedure into solving problems. Sometimes it's an inductive, you're making observations and you're predicting about what generally goes on in this phenomenon. Or it is a purpose. You're looking at all the variables and you're determining what is the most reasonable, so you're reasoning from the general to the specific. So you can use both types of logic in these labs depending on your goal. So my first introduction to the classical method of teaching was Dorothy Sayers. My wife brought the book home when we were doing classical conversations. I read the book. And I was like, yeah, I kind of see that in the development of a child, that it goes through, you know, this grammar phase into the dialectic and the logic and into the rhetoric phase. But I really didn't understand the 7 liberal arts that much. I got a pretty good education last night in the pre-conference with Dr. Rapinchuk. And a lot of these 7 liberal arts, there were actually 9 at one time, whittled down, that it is a process of learning. So I was kind of vindicated because I'd already started thinking that the trivium would be the grammar, the logic and the rhetoric, is a way of learning systems. Now, here at the College of the Ozarks, your first year, after your first year of teaching, you have to do a faith education project. It's a thesis. So I collected all this information about sciences, trying to teach modern sciences in a classical approach. And I found that this systems thinking is a new way of coming to this process of learning. It's a new face on an old thought. And the old thought is the grammar system. So what is that? In the grammar, I consider grammar like the parts. I'm going to use systems and concepts back and forth. So don't get confused when I switch back and forth. But I think of things more like systems. For grammar, talking about cells, first thing you got to do is, well, what is a cell in relation to something else? What is a cell made of? Then you start looking at all the components of a cell, and then you start looking at their interactions. Once you start with the pieces of the model, once you know the pieces of the model, and you start seeing how there's a cause and effect relationship or energy flows to make movement coming from kinetic to potential, you're starting to hook together the logic of that system or that concept model. So that's how I consider the grammar is just the pieces, the parts. And then the logic is how energy flows through them in a cause and effect manner. And then the rhetoric is how can we test this system, this concept model that we've got in our classroom. Now I'm going out on a limb here because when I came to the classroom, I had this idea of the grammar, the logic, and then the rhetoric. And how do I get my kids, my students, into this grammar phase? And my first couple years of teaching, I would start off and I would be using language that was unfamiliar to them. And I didn't quite understand that you've got to find out where they are in the process of their understanding of whatever concept, like cellular respiration is the one I'm going to use as an example today. Where are they at in that? What level of understanding do they have? And so when I learned to connect at that level, then I started to say, okay, now we're going to need to start framing. Now that I know where you're at, we're going to need to start framing a new or more intricacy into the model that they already had, fleshing it out. So I just started calling this concept framing. It's the way I get to grammar to logic. One of the first things that I do once we start into a new phase, a new concept, a new chapter, is I'm not even going to start lecturing at that point. They have a science text, and one of the things that my wife realized early on is that the kids coming out of public schools to her classroom could not read a science text. They could not read it to ascertain what was important and what was not. So the first thing that we do, and we got some pretty good texts, we use 9th grade Miller and Levine. It's not too heavy on the materialism. In chemistry we use Zumdahl. Not too much materialistic mindset in that one at all. But in AP biology, that's the Campbell text, it is infused with materialism, and we have to deal with that the very first of the year. We look at Christian worldview, and we look at material worldview, and we look at 4 fundamental questions that each worldview has to have, and they would quickly go through, or not really quickly, but we go through each one of these questions and compare and contrast what materialism has to say about origins, about meaning, purpose, and then destiny. And then we contrast that with the Christian worldview. So as they read through these texts, there will be certain times that I say, listen to what the text is saying here. Now, that is a material faith statement. There is no science in that. That is how they view the world, but no real proof. It's a faith statement. So that's how we start. But I send them into the textbook, and I have a glossary sheet. And sometimes I say, if there's certain texts that will not give a good definition, do you know what I mean when I say that? They'll give a word in bold, but they kind of generally describe it, but they never really come down and articulate that term. They never populate it with some sort of absolute type of description. So if there's components of that, I send them to the glossary. But mainly, I want them to pull those out of the text. And we use a Cornell style. I give them, I print them out. They've got lots of room to where we're going to add to this when we do our lecturing conversation. So I send them there, and that's the first step. The next one that I am a visual learner, and this is an example of a concept map. So this one is one of my seniors. I didn't ask her to do this level of detail, but when I saw it, I was blown away. This is by Victoria Smith. And this one is from AP Biology, and this is membranes. And I go through and I make a list of all the things that I want them to draw. Now, we haven't even lectured at this point. Because sometimes if you start lecturing before they've got enough framework, you're not adding to them, they don't have any connection to it, and then they just zone out.
Have they read the text at this point?
Yes.
Okay.
All right, so they've read the text, they go through, and now they start drawing these. And I have found that this, I will pass this around, but sometimes when you pass things around, people stop listening. But I just think this is the greatest. And she even made these little hidden panels, right? So you open the panel, and there's the type of transmembrane protein carriers, and which one was active and which one was passive.
Could we take a picture of it later?
Absolutely, absolutely. The bigger, the better. You know, and then I even will make a slide on the slideshow with all the things and the proportions I want them. So they draw them, and I say, okay, now from that point, there are, a lot of times the text will be written out to the side, but down at the bottom is the description of the illustration. I said, now read that, condense it, and then put it in where it belongs in your concept map. And as they go along, so here's the challenge that I'm trying to get. Well, when I'm working on a car, I know that car is three-dimensional, right? And so I can start diagnosing it, and I can get around it, I can get up in it, but we can't do that with a cell. We can't do that with polyatomic molecules and actually see things come together. We have to use some sort of imaginary device. And for biology especially, this gets that started. So I'll start asking them, show me where the energy and the movement is in that map. I mean, a lot of times you will have arrows in a metabolic pathway, either catabolic or anabolic. Those arrows are really deceptive because those arrows are hiding the fact that there are protein machines on this big long assembly line. They're biological robots with software that runs them in the background. So we talk about what those arrows actually are. Sometimes I'll have them line up, and then I have a puzzle, and we start at one end, and they're all proteins, and their hind ends are embedded into a phospholipid membrane. And then they have to receive the piece, and then another person brings the piece to them. Those will be your motor proteins. And so we physically go along and we build this puzzle, and at the other end, we'll have somebody receiving it, and I'll say, okay, that's a kinase. We're going to take that, wrap that in a vesicle, and take it outside, and we've just produced a product like a cell. So we get at this three-dimensional through the concept mapping.
Is that an example of experiential teaching, what you just described, or am I jumping ahead?
I believe it is, but I'm still trying to come up with my own definition of experiential teaching. That was what I was going to ask you guys.
Sorry. I would say that.
All right, so yeah, so experiential teaching. What do you think that means for experiential teaching?
Well, my first.
It's fun to say.
My first thought when it just popped up was that it was going to be the students, because it says student-led.
Right.
So I was thinking, oh, it's going to be the students experiencing teaching, letting them teach.
Now, I do flip my classroom with the seniors because they can handle it.
Well, that's really great, but.
Especially, you know, they come out of a full year of basically Chem 1, and we jump into biology, and basically the second chapter is always chemistry. And I'm like, I just spent a year teaching you this. You tell me what you know. So.
Well, when I think about the work it takes to get a lab ready for, even I have to set 5 this year, it's so much work and all this hauling around, and I think.
Linda, I will be praying for you.
Oh, no, thank you. But
Yeah,
I mean, it's a
it is a deal. And cleaning up is a deal.
All of it, right? And I think,
Yeah.
what if I just said, look, you guys do it all? Not because I don't want to, but because that's the sure way to make sure they understand that they're completely in charge of it.
Then I thank God, because I think that I can help you today.
Good.
One of the things I asked for my daily bread today was that what I could do would be a help. So I think this is going to help you. All right, experiential teaching. Anybody else got a shot at that? All right, so yes, ma'am. Yes, ma'am.
When they actually do something and you see, like, oh, that's how those things work. Like, I teach math. I used to teach science also, and I'm an engineer, so I got that too. But when, like, I have them do constructions, and it's like, we're gonna divide a line into 3 pieces, and this is the process. And then, can you see what you would do to divide it in 4 pieces? What about 5 pieces? And that, when they see that, it's like, oh wow, the segments really are the same. And we follow the directions. Wow.
So before I left the Park Service, I was the education outreach, and a friend of mine, who I'm going to credit here in a bit, and my wife and several others worked on conservation education. And when we had a stream ecology lab, so the first thing we would do is we would ask these students, we talked a little bit about dissolved oxygen in catfish and temperature needs. And there was a pond outside of the nature center. We had a canoe, strapping tape, a multi-probe that did dissolved oxygen temperature and a depth. And we basically asked them to do a cross-sectional profile. You know, we described it a little bit, but we just kind of threw it into them. And there was a lot of fussing and like, well, I don't know. And I was like, I've got mud. And it was just a whole thing going. And so we'd slowly give what they needed, you know, but we made them, they started the mental process of trying to visualize depth integrated transect, right? And so we would help them along the way. That beginning struggle creates all kinds of endorphins in the brain that activate learning. Because we're trying to create new neuron pathways here. That struggle actually produces the wiring together of the multipolar neurons in the brain. What they do, and that encourages long-term memory. But you've got to start them with the struggle just a bit. But students aren't like that. They're not used to that. They're like, spoon feed me, spoon feed me. So in the classroom, my friend Nathan, who I'm going to cite here in a minute, I went to one of his programs, and it was just, we started with, I forgot what we were reacting. We measured in grams, all the reactants, all the apparatus that we built. We put a balloon in there, and then we dumped the powder in the balloon. He didn't even tell us how to do that. We had to figure it out on our own. You know, and it was very vague at first, and it was like, where are we going here? Then we weighed it all again, and it was like, what's the observation? Well, we've got a chemical reaction. This reacted and created a gas, but it weighs the same. Conservation of mass. So that was my first introduction to experiential learning. I can do this a little bit in the classroom, but I've got an hour and 20 minutes, and I'm looking at the clock making sure that I'm not going to do it again, that I run to the end and then we're still not done, you know. So experiential learning, homeschooling, if you just schedule out your time and stay with it, it's a better model. I mean, I wish that there was like, oh, we're having Tuesday's Science Day. We're just doing science all day. And you can do math and geometry on another day, and I've got you all day, and we're just going to go through this process. But it's not like that. So the lectures at that point start to already become a training tool. Usually start my lecture with little quizzes, like words behind a block, or an illustration or concept that they were supposed to draw with blocks over it, and they have to tell me by groups. We have houses here, and then I also assign research teams, 25 in a classroom, so I've got sets of 5 on everything that I do. It turns out we have 5 houses because there's 5 days a week. Worked out really well. But it turns in more of a discussions, because the students that are really into this, ones that are really pursuing knowledge, will have questions at that point. And sometimes the questions can actually just, I can put the slide clicker down because we just covered the last 3 slides. I don't have to show you that. You've already known that from what you've taught yourself in the concept building phase. What happens?
Your slide went off.
Okay.
We don't have.
I think we just have to turn it on and off.
That gives me time to hand this out.
Here, come here.
Oh, thank you.
So you would do this experiential teaching prior to doing a lab?
Yeah. Yeah. The lab is where we explore the rhetoric level.
Okay.
They have to use what they know in the frame of the scientific method process to answer a question. So they have to, not only that, they have to, where's the tool talk? They have to have a tool talk. And it might be as simple as, I forgot my graduated cylinder, well, it's got one over here, is how to use a pipette. Because the question I'm going to ask them, are probably going to need to know what a pipette is and how to use an automatic pipetter. This whole thing over here was we're testing in 12th grade the yeast's ability to metabolize different forms of sugar. So we got dextrose, which is a glucose and a water molecule. We got 2 disaccharides, and then we got amylase, which is cornstarch, a huge polysaccharide. At that time, I try to use what they've learned in chemistry already. So if it's possible, I make them make up a molar solution of equal concentrations. But in this case, since the amylase, who knows how much actual molecules are there, because it's in bits and pieces of a long chain, we just did percent solution. All right, so they had to figure out how to keep things equal, percent solution. This is a gas trap. They had already used this in photosynthesis, so they already knew what that was about. There's 2 forms that I ask one team to do. Is, okay, I want you to only look at the side of alcohol fermentation. So glycolysis, cut off the oxygen, and see how much CO2 gets produced. And then other groups had something similar open to the air. We don't have any way to infuse oxygen into it. But it was an open system, so it was a combination of alcoholic fermentation and cellular respiration, because yeasts are facultative. They could do both. So, and then the fermentation tubes. So we would talk about that. We'd talk about how to make a proper weight. You know, a solution. Be sure that you add how many grams of solute that you're going to put in. You got to add first, because if you try to get 100 milliliters and add it in, you got 105. Now that's, your technique was bad. So the tool talks is just, if I anticipate what they will need, I will preface that, or in the lecture, I will bring something out and show how to use it. So basically, they have all the ability, they have the knowledge, and they have the ability. And then that gets us into where we play the game. All right, so the students are already divided in teams. We have houses. I'm thinking I like the houses a little better. We're the first year with that. Every time we do, the very first one that we do, make sure I get out of whack here. Did everybody get one of these? All right. The very first lab is nothing but practicing this process. Because you have to practice it, they have to understand the rules of the game, you have to lay out your expectations of what you expect them to do, and give them the parameters to play the game. So the first time I do this for my 9th graders, it's something very simple, experimental design, very simple, and we're just going through the process of this type of lab. The guiding question. For photosynthesis, a guiding question could be, what is the effect of 2 different levels of light intensity on photosynthesis? I can have one group do that. I can go over here, we'll say, okay, we're going to put in sodium bicarbonate at a 5% solution in this and a 10% solution. This team over here, you tell me the difference, the effect that that has on photosynthesis rate. And then I might go over here to this team and say, we're going to do this, but we're going to only use this type of aquatic plant and keep all the variables, and we're going to use this type of aquatic plant and ask which one has the highest photosynthetic rate. Or like in this model, we just said, okay, this team over here, you're going to pick 2 sugars. You guys are going to do 4. This team over here, you're going to do with oxygen and out oxygen and looking at the variable of gas production, CO2. And that really helps too, because when they actually do present their data, it's not the same as what this team did. Because I've been doing that, but the people in the back are going like, I see them scurrying around. They're making last minute adjustments to their proposal or their presentation. All right.
So the guiding question is not universal to the...
If I can help it, I will, and it all depends on what equipment I have in my shop. Mm-hmm. What tools I have in the toolbox. And in chemistry, I'm very limited because I don't have very good scales. Yeah. So when you start looking at stoichiometry and predicting product yield, you don't have a good measurement on what goes in. You're surely not going to get a good measure. It's not lunchtime yet. Hang on. Nobody move. That bell scares me, right? I've been trained by that bell to do something when it rings. All right, so guiding question. Where are we at? Where's the background part? Have I... Proposal development. Where's my background? Oh, it's down here at the bottom. All right, so that's one of the most important. Maybe I should take this and put it up here. I asked the question, I said, all right, if you're going to write this up in your lab manual and you're going to share it with a friend or your parents, you have to show them, tell them enough of a framework of information that they could determine whether you have found a proper answer or not. So the background is pretty important. We go to the text, and it could be up to 3 to 4 paragraphs on background. Have them cite the text. Sometimes if I can get them, we'll go to the computer lab. They will pull down some sort of article from the computer lab and cite that. So the background is really practicing trying to come up with ideas that frame why your question is important. So it's down there. Then that leads us into cause and effect. So cause and effect and energy. There is no movement without energy. And there's no movement without cause and effect. It is the threads that go between any type of scientific endeavor. In the public school, those are cross threads, or what are those called? Crossover themes. Integration. Yeah, something like that. Well, truly, this is even, you could even go cause and effect all the way back to argue the existence of God with these things. I mean, and then energy of itself, what is it? We can characterize it, but its essence is unknown. It's pretty mysterious stuff.
Can I ask a quick question?
Yes, ma'am.
It's exact. I have this. When you were showing us the concept framing and the creation of the map, you said you would pose the question, show me the energy, where is the energy or the movement in this concept? I was going to ask then, and now I want to ask it now.
Okay. Yeah.
Is that a universal question that you would ask every time because of the idea that there's there's always energy and they work together.
Yep.
So it doesn't matter what they're drawing, that's always something to look for.
Right.
And then, okay.
Yep. It's not, you know, that in chemistry, we never leave it. Activation energy.
Yeah.
So you got to have motion. Those two atoms have to smash together at some rate of speed in order to get things to happen.
Yeah.
You know, so where did that kinetic energy come from? I mean, you're adding heat to the system to causing it to move to that speed. But they're not able to give a good discussion or conclusion if they do not understand the step-by-step causal effects. And that energy is driving that step. So we have those conversations over and over again. Kinetic to potential, all right, so we're in photosynthesis, that's the kinetic movement, that's the ambient temperature, but it's also the light coming in at a speed, hitting that, absorbing that, causing that electron to jump to a higher state, move down the electron transport chain, pump hydrogen ions across membranes, that sort of thing. So all that action, all that energy, especially when it comes to cellular respiration, those redox steps all the way around the citric acid cycles are discussions where energy, what is that? What is the energy in that electron? The more we have discussions, the more I go back to, we've got to really have, that Campbell, all right, that's a pretty in-depth book. I go back and reread that and I'll go like, I'm thinking I'm getting this a little deeper. So, well, I mean, that challenges me to push a little harder because apparently my concept model is not as detailed as it needs to be. So I'm still working on mine. Experimental variables drive them nuts, trying to figure out which one is which. So a control. But here's the thing. Whatever we're manipulating is the independent variable. Over here, we are manipulating yeast cells by exposing them to 3 or 4 different types of sugar formation and even isomers of each other. And if we can measure the dependent variable, and by the way, this started this morning, this is sucrose, just table sugar. There was one little bubble here, and I don't know how many milliliters I've got going there, but we could do some stoichiometry on that. I mean, we can't calculate, since I don't know barometric pressure in this tube because it's on a vacuum, and I don't know its temperature, I can't calculate how many CO2 molecules we have in there, but we can talk about how we could make this more efficient to do that. So, I mean, you can get in some really good conversations. It's very helpful in biology if they already had chemistry, because it's all what is biology. It's just various levels of chemistry interacting with each other. All right. Now look at the science proposal, the writing guide that I gave you. For 3 years, I have worked on this, and I'm still not satisfied with it. So I'm going to discuss it with you, and I've talked too much already. Hey, by the way, we're going to be sitting, if you will, sit with me at lunch, and we can just continue this conversation if I continue to talk and run over. So go all the way down to methods and put your hand over that. That's the proposal. Now, I took this writing guide or rubric from the College of the Ozarks. And then I looked and I found 5 or 6 different rubrics on the internet, and I said, what do I want? How do I want the expectations to be laid out for my students to attend to? So the introduction, beginning somebody's going to have to read this. Put some art into it, right? Let's put some flair into it. Let's make it interesting and engaging sentence. But before you're done with the introduction, you're going to have to state the purpose. Now, I'm already better than the labs that I grew up with, because you didn't know the purpose until you were working on the post lab, and you're going like, that's what we were doing the entire time? So before we even start the lab, they have to tell me the purpose. The background, that's information. I can even pull up slides, and we'll copy down slides from the lectures or something, or pieces of quotes out of the book. We'll lay that down. And then at the very end again, they have to tell me the purpose. Now, I say hypothesis and purpose. There's where we get in conversation as to what is inductive logic and deductive logic. if we have a test, a hypothesis where we've narrowed this down to this two things can happen, one thing can happen, and then we're saying something very specific and we're making a general statement about all photosynthesis, then that would be inductive. Now, if I'm asking them a question like we've got a blood typing lab coming up where they don't know, the parents have died, we've got 3 kids, and the parents over here, one of them had died and one of them's unconscious and can't speak. We've got their blood, and as responders, emergency responders and scientists, we have to determine who these 3 babies belong to. Now, that's a purpose. That's going to be using forensic deductive. And we have to understand which type that we're using on that. And then, if it's a hypothesis test, we're going to need some sort of control variables, QA/QC. The writing guide, now they're working as a team here. So all the way from introduction to methods, of the 5 kids on that team, if they have very similar text and writing, I'm not worried about that. Some they don't like to copy off each other, some want to do more original. All right, so the hypothesis statement, methods, they have to give me a list. And then they put it in their tubs. And I'll come and say, all right, I'm going to put whatever you say in the tubs. And I'm knowing that they're not going to get it all and they'll need some more later on. You know, I almost thought, well, I'll say, well, that's going to cost you. You know, you got to owe me, you have to clean the whiteboards or something. So I fill their tubs. And sometimes I put in stuff like a Snickers bar or Airheads or something like that. And I usually just put in a cough drop because that's what we can give them. Let's see.
This is a list of the procedures?
Yeah, the materials.
The materials.
They've already got the procedures. Now they're giving me materials. I put under methods, I have procedures and materials. That way they don't get those confused. And we do this the same way every time. Then we have the lab, which is usually fast and furious. There has to be some sort of safety talk. Because in the classroom, there's only certain level of chemicals that I'm going to be able to use. If it bumps up, if there's any of this is flammability, reactivity, and health. If that bumps up to a 2, I got to go to a different setting. So I'm just now getting through my chemical inventory, thanks to Shinani, and determining what I actually have in my classroom. And quite a bit of the stuff I've integrated into our chemistry department. And then what they need to wear, what level of PPE to mitigate personal protection equipment to mitigate whatever hazards in the room. And even if it's a class 1, we were doing a BTB solution at 2%. It's a 1 on, I think it was on reactivity, or was it health? Anyway, I had one student blow through the straw. She took a sip. All right, right in the middle, I was like, duh! You sipped, everybody's blowing. I showed you, you know, so it happens. You have to have those SDSs, and you have to have something in place. You got to call parents and start trying to determine, is this a thing or not? So that's when I just have a stress meltdown. I just probably crawl under my desk and don't even go to lunch. And it's like, all right, if the cops come, I'm not here. I did my best. All right, so I seriously, that worries me to death. I've had cancer. I've had students in my room who've had cancer. I've had students who had parents die of cancer. I don't want to expose anybody to anything that they might be susceptible to and I have no knowledge of. So that does scare me. They will have problems. Seniors, I don't give that much guidance to. And they had some problems. They chose to do a detailed measurement using this dependent variable of change in balloon. Well, I saw it was going to leak. I saw, is that balloon symmetrical to you? I mean, that's going to be absence or presence. That's not a really quantitative. So I just let it go. And then the next morning I come in, their balloons were all deflated. And they were like, oh, I'm like, you don't have any data. Well, maybe I've got some resources. Maybe you should do that again. Maybe you should try a different approach. Because they had multiple approaches they could try, but they were kind of being lazy. So I'm just like, we'll just let that one go and see where it comes. Data collection, and then they start developing tables and graphs. Here's where they struggle quite a bit. is how do I make a table where the independent variable and the dependent variable, I don't get those mixed up. How do I make a graph where the x-axis is the manipulated thing and the y-axis is the measured thing? And so we go through that time and again, and sometimes they invert them on their whiteboards and we won't tell them until they present. And some student will go like, yeah, isn't that your independent on, yeah, and they're like, ooh. So we're back there making notes. All right, as soon as the data is collected, they start the whiteboards. Now we're getting into the rhetoric level. They'll start creating. This is a lot of fun, and I know that some of the girls can really do a very good artistic rendering. I mean, these are high quality. There's only one gal on this team, but this was a really good study. It's laid out this. They have to say the guiding question, and most of the time they leave their claim blank until they assess the data. They collect the data. This is the evidence box. This is the justification, or we call it the logic. In 9th grade, I try to get them to boil it down to if-then statements. And then they can pull in, they can put in a small box of their background, just a little bit of bullet points. So they lay out their evidence here, and it has to be something that's simple enough that the rest of the class can see it and understand it and get at the point. And sometimes there's enough room on there for a table. And then on this side, like I say, I try to make them use an if-then type statement, forces them back to the 8th grade logic that they're probably trying to do a data dump on all the time. So the idea is that when this leads to this, they make a claim, and when these things are in union, there's no contradiction between their claim, the evidence, and the reasoning, then they have just formulated a scientific argument. And I have a scoring matrix for the board. Now, I'm going to have to say, if you write up on this scoring matrix for the whiteboard grading, the kids grade each other. The teams grade each team. I have the final say. And I've got this big spreadsheet where I put all the data in. It does an analysis or a sum, and then it has a printout. I just made that up. I looked around. I could not find something that was suitable from what this need was, and I needed some sort of score on this verbal presentation. So this is their first go at rhetoric, right? Now they've learned the system, they've manipulated it, they know the logic of how it works, they've manipulated it, and now they're making a claim about it. And then they have to defend that claim, and sometimes, especially now that I've rearranged it to houses, there's a pretty good question because they want to see which house is going to be the best. And then other grades are in that house and want to know how they did, so that's working out pretty well. That's the scoring matrix. If you see on the scoring matrix, I got it pretty much memorized. It's only 1 through 4, and it's not detailed enough. I think it needs to be like 1 through 8, and it's not a good product to tell the students what they did wrong. So I don't like this rubric. If you've got a way, if you read that and you want to see, you think you can make that more distinct and where the feedback is more meaningful, if they're presenting like this the entire time talking to their board, there needs to be some way that needs to be corrected. If they're trying to be cute and witty and it distracts from what they're trying to do, there's somewhere it needs to be, and I've still yet to find something I'm satisfied.
But you created this, right?
I looked the internet, I went pieces and parts, I rewrote that, and at the end point I said, I have got to go with something at this point, but I want to.
The hard part is once you start with it, you have to keep going with it. Otherwise you keep taking it for a while anyway.
Till the end of the year.
Yeah.
Which is coming pretty quick.
Yeah.
Now this I'm keeping, and I've even had 9th graders say, tell me what's confusing about this. And they'll go, I don't understand this, or I don't understand that. And so I keep changing it. Now, I don't want to bring it down to their level. I want to set it just out of reach a little bit where they have to go for something higher than that.
Do you have an example of one of these that's done? Like,
I've got a picture of one.
okay.
All right, so now in the report, in their lab notebooks, they've already got introduction through methods. So after their lab, the results, if you look on the guide, the results is basically this. They have to put their graphs and their charts in the results, and then they have to describe it, what's going on in that graph and chart. Now, this is not interpretation. This is just statement of facts in a chronological or logical order. I don't want any interpretation going on here, because interpretation goes in this section, the logic. That would be in the discussion. That's where you bring in cause and effect. That's where you bring in what happened in there. That's when you start interpreting the data and into the argument and fashioning the argument in the discussion. The conclusion is basically wrapping up what you've started with. You need to, whatever statement you started introductory needs the very, very last part. But basically a synthesis of this discussion quickly. Just a review and a reiteration of what was in the discussion boiled down. Still they get that confused. They'll put interpretation up in results and then start statement of facts in discussion, and it's really hard. I started them out, I just said, okay, what's the standard? Go running out of time?
10 minutes.
10 minutes? I think I can wrap it. I was having them draw in pen. I said, after this year, I said, no. Just do it in pencil. And between sections, give yourself about an inch or so space to where you're going to add stuff later. You're going to be able to reread what you did and make some changes. All right. Then we go, once it's done. Now, I got this guidance from the National Science Teachers Association, and I have the books, Argument-Driven Inquiry. I used one of these labs and then immediately started changing them. I think it is, classical works, and so they're going back to things like that in the public school system. They're trying to push those things in public schools. And my pastor teaches, he's bivocational, he teaches 7th through 9th at a very, very poor public school in Arkansas. And he's trying this, but those labs will take him up to a week and a half. He's like, I got curriculum we got to go through. So he's already, kind of said this is not going to work. But they're trying to make these type of things, trying to teach classically, basically, in some ways. All right. But they recommended a double-blind peer review, which would require it all being done electronically and typed up, and which causes all kinds of issues as to when they're doing their background, they just go and cut and paste, and I have to go and use Turnitin and try to keep everybody from plagiarism. If they're writing in their journal, in their notebook, there's just not much of that. And then I'm usually there with them in the writing process. I've substituted for a student review. So gather everybody's notebooks with one of these in the notebook, and then I just hand them out to students that are on a different team. And they have about 20 minutes to read it and edit it, and then I say, please, do your fellow student a favor and pursue excellence in your editing. I'm looking for a way to grade their editing skills.
Grading or grading.
Grading their grading. I'm looking for that. That would be my next way to really force them into helping the writing process. Because when you got 25 students, you can't do all those books yourself. I mean, just forget your weekend. Forget anything you're going to do with your family after night with those. So this is a way to cut your time down and put the experience of editing back into other of the students' hands. So they write their comments. This is some of the ones I wrote up. I just circle stuff, and I have them circle stuff, and then they will write comments, and they will score this. Now, I just use a ratio of the score from this, and my labs are usually 20 to 30 points. Depending on the complexity, I scale it back down. But they will go ahead and score. And then they will turn that back to the student, and then I allow them time to fix it. And even if they have questions, they fix it. Unfortunately, most students just say, like, well, I like that score. They turn it in. So I usually get after them pretty good on the editing. But those, you can see the erase marks, you can read what the student read, and you can see the changes, and you say, okay, this student was conscientious about trying to pursue excellence. That's where we go with that. And then I put my own comments on that as well. And then they get a score and it comes back. We laid the grammar down, concept map. We got them to where they're comfortable manipulating the system, the model. And then rhetorical skills. They did their presentations. And they have to stand up. And they get nervous. You know, they talk at each other all day long, but as soon as they stand up at the board, well, how many of you guys, all of a sudden you go to, and you find out you can no longer spell? It's like, how does that work? Whatever. Okay. Just spell it really sloppy and they don't know. So that's that process. Okay. We have to practice these labs for them to get used to the rules of the game. You use the scientific method again and again and again, and assign variables again and again. You assign what is control again and again, and so they get used to just finding out, okay, this is manipulating, and then really you're teaching them how to do the science method, the process. The repetition on science writing. Over and again, it held to the same standards, using different labs, but reporting it in the same way. I've wrote about 30 reports for the federal government in my time, and they all use this same pattern. All right, so it's not a mysterious thing. They will see it again if they pursue science. Downside, this takes a lot of time. This can fully eat up a week. But if it's a good thing, if it's something that they will go like, hey, I remember that time that we blew up the balloon and spattered stuff all over the science room or something along those lines. It's not a good example. Probably shouldn't use that. Anyway, they will remember these things.
The mistakes they make, they remember, oh, we had to do that 3 times.
Yeah.
That was bad.
As long as they don't fixate on their mistakes, they learn to go past them. So how I do this, if I'm in a rush, I just cut it off the whiteboard. I still got a score from the presentations, and then we move on. That cuts down a period. Yes, ma'am.
So I love this concept and everything, but like timeline-wise, do you have an hour for a day, 5 days a week, or what do you have?
I have 2 block periods. So what is that?
Hour and a half.
Hour and a half.
Okay.
I can do most labs in that block period.
Okay.
Remember, it's got to be done in my classroom. I do take chemists over there to the chemistry department. I'm pointing at the building where it's at. But it takes 10 minutes to get over there, 5 to 10 minutes to set up. All right, and then I have to go over there beforehand and get permission and everything, and they're great over there, don't get me wrong. But we only have like really 40 minutes of lab time if I use that lab. But if we immediately get started on something simpler, I can use my classroom. So I'm trying to come up with the materials that are not hazardous in any way, because kids eat in my classroom every day. So we got to be super careful with that. But it's a way to speed things up. Now my seniors, I only have them for 1 period a day. So it's basically, we just cut it into chunks. And it's super hard to have any meaningful lab in that time period. So, but we did get something like this done. And these guys over here, they tested 3 different types of sugars, both with and without oxygen. So they did a split level design, which I was pretty impressed with. Where are we at?
Excuse me. When you say stuff at the whiteboard presentations, do you mean to not do the report?
Right. But I got to know ahead of time. Because if they go ahead and start the lab reporting process, they need to finish it. So it's like this next week. I've got 2 labs in chemistry. There's no way to do this process. We got spring break. Have to wrap up that chapter. I can't leave it open during spring break, so we're not even going to do this lab. We're just going to do a demo. So it is mix and match. I want to give them acknowledgments. Here's my mentor, my wife. The love of my life for over 30 years of marriage. She's assistant professor, soon to be associate, at North Arkansas College. She guided me through my mentoring process, and she helped me a lot with, she's like, hey, I can't do this in my classroom, but you can do it in yours. And then Nathan Wendell is a science education specialist. I met him years ago doing conservation education. He taught at public schools, but he was classically trained. And he's the one that brought this system to me. And this is this National Science Teacher Association. I don't think it costs anything.
When was that published? First?
I'm not for sure.
20 years, probably, or-
No, it's not. It's fairly recent.
More recent.
Fairly recent. And the reason I don't call this argument-driven inquiry, the second page on that, they copyrighted everything. The language they copyrighted. It's like, you can't copyright the science method.
Right.
So, and plus, I kind of left their methodology, the framework I still use, but I left their methodology like the second lab, just because it wasn't going to work. Wasn't, you know, if you're homeschooling though, you can stay with a topic a lot longer. And so, yeah. So I still meet with Nathan once a month or something like that. To try, he and I are trying to work and collaborate on getting a library of experiential learning opportunities for chemistry and biology. I'm trying to get him to publish it through Canon Press. He's an open source guy, and I was like, well, that's nice, that's kind, but the people I'm visiting with probably won't Google and find your site. You're so targeted on public schools, because it's who he works with. He's got 16 school districts he works with. So I'm trying to get him to do that with me. He said he wasn't opposed to publishing it, but we're working on that. So that's what I'm going to do this summer, is to try to find how many things I can put in my teaching library, my toolbox, where I can change the classroom environment from passive to active and walk them through the concept building instead of lecturing at them, which is what I've done to you today. I tried to come up with a way to do this in an hour, and I just said this is the best. So, questions during lunch? We sit at the same table?
We
Yes.
all want to sign up for lunch.
Okay, yeah. There's should be, our tables should be designated.
Okay.
Yeah, because
Oh,
we're all going to be eating in groups of what we do.
Okay.
And not from, because last year we noticed that we were all eating together and you guys were all, and so we're trying to cross-pollinate here. And if you want to see cross-pollinating, there's bees in my classroom. All right. No, like really, there's bees in the classroom.
Thank you.
All right. Now,